Okay, everybody, as promised, I'm putting a lecture up on YouTube for us, and I'm just doing a little screen capture lecture for you all so that you can see the notes here. But we are picking up where we left off from today's lecture. So we are moving on to human genetic disorders. So today's topic is human genetic disorders. Okay, we've talked about a few traits that humans can inherit. We've talked about polygenic traits like eye color. We've talked about straight up Mendelian genetic, the inherited traits such as what your earlobe looks like. We've looked at codominance, which is where we left off with blood type. Human blood type exhibits a codominant inheritance pattern. But when someone has a baby, no one ever just thinks, oh gosh, I hope my baby has the same blood type as I do. No one cares about that. What we care about is that we have a healthy child. And there are numerous genetically inherited traits that can cause physical problems for people. So human genetic disorders refers to those traits that we would rather not pass on to our kids, but sometimes we do. Human genetic disorders fall into generally one of two categories. You basically have what we call autosomal disorders versus what we call X-linked disorders. Now, as you may or may not remember, an autosome is a chromosome that is not a sex chromosome. So it's one of the numbered chromosomes. So autosomal disorders are caused by a faulty allele that is on a numbered chromosome. So these are disorders caused by a faulty allele that is on an autosome, non-sex chromosome. There we go. So you could have a faulty allele that is present on an autosome. That type of inheritance pattern is going to be different from an X-linked disorder, which is when a disorder is caused by a faulty allele that is on the X chromosome. Sometimes these are called sex-linked, but we're only talking about alleles that would be on the X chromosome here. So sometimes there's a faulty allele on the Y chromosome, but they're just not that common because the X chromosome is so much bigger than the Y. If you remember from a couple of weeks ago, we talked about that. So X-linked disorders. This is when the faulty allele is on the X chromosome. The inheritance pattern is going to be different than an autosomal disorder. So we're going to look at autosomal disorders first. If we have an outline here, A, I have B over here. Autosomal disorders come in one or two varieties generally. And the first step would be what we call autosomal recessive. An autosomal recessive disorder is when the faulty allele is recessive and it's on an autosome. Faulty allele is recessive and on an autosome. There we go. And an example of this is a disease called cystic fibrosis. Individuals with cystic fibrosis are lacking a protein that allows certain ions to cross in and out of your cells. And what happens is that they end up with a lot of lung issues, a lot of sticky thick mucus will form in their lungs, and so they end up with a lot of lung infections. Many years ago, those individuals with cystic fibrosis did not have a very long lifespan. Medications have improved greatly since then, and actually there's a really interesting article, and if we had more time in the semester I would have you read it, about gene therapy for individuals with cystic fibrosis is finally starting to have some success, and this is the first instance of gene therapy actually working. And we have been working on gene therapy for 30 years now in the scientific community, and it looks like it's finally becoming a possibility. And we've had the most success now with individuals with cystic fibrosis. Gene therapy is replacing the idea of replacing a faulty allele with a healthy one, which doesn't happen on its own. But anyhow, individuals with cystic fibrosis live much longer than they used to, and it is caused by a faulty allele on an autosome that is recessive. So let's use, I don't want to use the letter C because a capital C looks so much like a lowercase c. Let's use the letter F for fibrosis. A dominant allele causes a normal phenotype. But the faulty allele, which is recessive, causes cystic fibrosis. So because this is an autosomal disorder, everyone's going to have two F alleles, right? And the possibilities are an individual could be homozygous dominant, or an individual could be heterozygous, or an individual could be homozygous recessive. Now think about it. Who's going to have the disorder? Which of these three genotypes is going to be affected with the disorder? You think about that? Well, I changed my pen color. Let's change it to, how about, we'll go with this orange color. And if you decided that homozygous recessive would be the affected individual, you are correct. So neither homozygous dominant or heterozygous are affected. These are both healthy genotypes. This individual won't have cystic fibrosis. This individual won't have cystic fibrosis. But what is the deal with this individual right here? The heterozygous person is what we call a carrier. A carrier is someone who has the allele for the disorder but doesn't have the disorder themselves. So a carrier has the faulty allele but not the disorder. So even though this is a healthy genotype, they are a carrier. This person over here, homozygous dominant, this genotype gives you a healthy phenotype, and they're not a carrier. They only have the normal healthy allele. Right, so let's go back to blue. So let's look at this affected individual here, this person who is homozygous recessive. Let's think about where they got each of these alleles. They had to get one from each parent, right? And so let's say that you're working as a genetic counselor. There are such things. You're working as a genetic counselor, and a couple comes to you who has a child that was just diagnosed with cystic fibrosis. And you need to find a way to explain to them that their child is homozygous recessive, And they had to have received one of these recessive alleles from their birth mother. Let's put mom over here. And they had to have received this recessive allele, the other one, from their birth father. So let's put the male there. Okay, now each of the parents, we don't know what their other allele is here. You know, we gotta fill in the blank there. It's a big question mark. We don't know what they have there. Let's get rid of those. All right, but the parents say to you, but how could they have how could our child have inherited this disorder from us? We don't have the disorder. What if the parents say, we don't have the disorder? The mother does not have cystic fibrosis. She's healthy. I can't spell healthy. There we go. And the father is like, I'm healthy too. And it turns out they are healthy, but they are carriers. So we know that this mystery allele right there must be a dominant allele. So both parents must be heterozygous to be healthy individuals and still have a child with the disorder. So here's something else to think about. What if this couple tells you, but okay, so we're both a carrier, we're healthy, that doesn't make sense because we have another child. Let's say we have an older child. Let's say this is child 2, and our first child, child 1, is healthy. Say, but our oldest child is healthy. So you're like, well, your oldest child, looking back up here, must be either this or this. We're coming up here. Eyes up here. All right. So child 1 must be healthy. So they are either this, which is a possibility between this cross, or they could be heterozygous. So let's say, as a genetic counselor, you have convinced this couple that they are indeed both carriers of this and they must be heterozygous for this trait. Let's say this oldest child, child 1 here, grows up and is wondering, I wonder if I am homozygous dominant or if I too am a carrier. Let's look at what the probability is for child 1 being a carrier. What is the probability of child 1 being I almost wrote character, a carrier? Right there. Well, we need to do the cross between the parents. So let's look at that. Let's look at this. Let's cross child 1's parents with each other. We know that they were both heterozygous. And we know when we're crossing 2 heterozygotes for one trait, all we need is a simple Punnett square box, 4 square box. Let's put this parent over here, can give their offspring either a dominant allele or a recessive allele. Likewise, this parent can donate a dominant allele or a recessive allele. We fill in the boxes. We've done this cross so many times, just with different letters. But what's the probability of child 1 being homozygous dominant or heterozygous? Or I guess we're looking at what's the probability of there being a carrier? All right, so carrier, this person's a carrier and this person's a carrier. And before you go saying, well, it's a 50-50 shot whether she's a carrier, and I'm telling you no, it's not a 2 out of 4 box shot. Okay, it was a 2 out of 4 chance that their parents would have someone as a carrier, but I'm saying that it's a little different for this child over here. I'll circle them for whether or not they are homozygous dominant or heterozygous. We can't say that there is a 2 out of 4 chance that she's a carrier, because that's not quite right, and I'll tell you why. Because out of these 4 boxes here, this box, this box, this box, and this box, we can eliminate one of those boxes. Think about which of these 4 boxes we can eliminate. I'll give you a moment. 1 Mississippi, 2 Mississippi, 3 Mississippi. We can rule out this box here. We know child 1 is not homozygous recessive because they don't have the disorder. We know the sibling is, but if we want to figure out what's the probability of child 1 being a carrier, we can eliminate this box. So there are only 3 boxes to choose from. So it's a 3, let's erase that. So it is a 1, 2 boxes out of 1, 2, 3 total boxes. It's a 2 and 3 shot of her being him or her. I don't know, I didn't say what the sex was. It is a 2 in 3 chance of there being a carrier. Child 1 is twice as likely to be a carrier than to be homozygous dominant. And that's something that they might want to think about before they have children. Today, you can just send a sample of your saliva off to one of the DNA sequencing services, and they can tell you right away which allele you have. That's definitely easier, but it's good to do this math. And before we had that available to us, this is what we had to do. We had no way of knowing, but we could tell someone that they would be twice as likely to be a carrier as not. So that's an interesting question right there. Autosomal recessive disorders. Right? Let's look at one more example of an autosomal recessive disorder. Let's go with let's go with pink for right now because we're talking blood. We're going to talk blood. There is a new example of, this is an example of an autosomal recessive disorder, and it's called sickle cell anemia. Individuals with sickle cell anemia make red blood cells that don't carry oxygen very well. Okay? It's controlled by a gene with a dominant allele and a recessive allele that's on an autosome. So the dominant allele, give it a capital S, causes normally shaped red blood cells. RBC stands for red blood cells. But the faulty allele is recessive, and I'll draw it like a script. I'll draw it as a script, lowercase s, so that we don't get our capital S's confused with our lowercase s, because if I do that may not work so well. So the recessive allele, lowercase script s, causes sickled red blood cells. A sickle is like a crescent shape. And so normally red blood cells are disc-shaped and kind of flattened a little bit, kind of like a Mentos, those Mentos candies that people were putting into Diet Coke all the time, kind of like those, kind of a flattened sort of disc. Whereas a sickled red blood cell gets kind of shrivelly and kind of crescent-shaped. This sickled shape does not carry oxygen very well. Okay, and you know what you need oxygen for. You need oxygen to get to all of your cells so that they can make ATP. All right? And I'm not going to get all into that because we've been there. This one carries oxygen well. This sickled red blood cell does not carry oxygen very well. Okay, so individuals can either be homozygous dominant, heterozygous, or homozygous recessive. This is the affected individual with sickled red blood cells. It's the same inheritance pattern as cystic fibrosis. It's autosomal recessive. But what's slightly interesting about sickle cell anemia are 2 things. The faulty allele, the recessive allele that causes sickle cell anemia, is more prevalent in African and African American, African anywhere populations. So, and here's the theory as to why that is. There's something very interesting that happens, and this concept is called pleiotropy. Pleiotropy is a concept that occurs when an allele causes more than one trait. So when an allele causes more than one trait to occur, I call it 2 traits for the price of 1, right? And what's interesting and why this recessive allele became more prevalent in communities that were established in much of Africa is this. There is something about being heterozygous for the trait. There's something about being a carrier that gave people an extra trait that was very advantageous in certain parts of Africa. Being a heterozygous gave you a lot of protection against developing malaria. So it also gave protection against a disease called malaria, which is an infection caused by a protozoa, and it's transmitted through mosquitoes that only are prevalent in certain parts of Africa. So it was evolutionarily advantageous to be heterozygous for the trait. So isn't that funky? And it's because of pleiotropy. So not only does the faulty allele cause some sickling of red blood cells, it also, at least in heterozygous individuals, gave them some protection against developing malaria, which is a huge advantage if you're living in an area that has that type of mosquito that carries that infection, that carries that little protozoa that when the mosquito bites you, transmits that infection into your blood. Malaria is a very, very serious disease, and people can die of it. People still die of it. So it's very advantageous to have some sort of natural protection against malaria. And so this faulty allele remained in populations where malaria existed. What's also interesting, so that's one interesting thing about sickle cell anemia. The other thing that's interesting about sickle cell anemia is that it exhibits some incomplete dominance. There is some incomplete dominance. So like in 4 o'clocks, how the heterozygous, that gave you pink flowers, right? This means that an individual who is heterozygous for the sickle allele, not only do they have some protection against malaria, but they actually do get some slight sickling to their red blood cells. So you get a blending of traits. Like over here, the homozygous dominant individual would have absolutely no sickling. The homozygous recessive individual, definitely sickle cell anemia. The heterozygote would have some slight sickling. Some slight sickling to their cells. So, the red blood cell maybe wouldn't look like this, quite like a crescent, but maybe it would be like that, something like that. But it still wouldn't carry oxygen quite as well as a red blood cell that does not display any sickling, like this one up here. I'm drawing arrows for you all because you can't see me pointing at my screen. Anyhow, so several years ago, gosh, probably like 12 years ago, there was a football player, gosh, I can't remember the guy's name, but he played for the Pittsburgh Steelers, and the Pittsburgh Steelers were playing in Denver. They were playing the Broncos, right? Denver is high altitude, and so there is slightly less oxygen in the atmosphere there. People who live at high altitudes will naturally produce more red blood cells in response to living there so that they can carry enough oxygen to their cells. But individuals who live at lower elevations, like sea level, or Pittsburgh's really not, I don't know what the elevation of Pittsburgh is, but it ain't very, very high up there, would not have quite as many red blood cells circulating in their system as people who lived and trained in Denver. Denver is known as the Mile High City, so that's what, it's over 5,000 feet. Okay? So this individual is exerting himself. Didn't know, this football player for the Pittsburgh Steelers did not know that he is heterozygous for the sickle cell anemia gene, and had some slight sickling of his red blood cells because, he didn't live at high altitude. He went to high altitude, began exerting himself, and as you know, when you're exerting yourself running, for example, or, running a ball, you need a little bit more oxygen, and he didn't have it, and so some of his cells were damaged as a result of that. The guy had to have his spleen removed as a result because he had some slight sickling to his red blood cells and didn't know it. So that's something apparently that athletes are tested for now. This fellow is not allowed to play at high altitudes anymore. Next time they went to Denver, he was benched because it's harmful to his body to exert himself at high altitudes like that. Lower altitudes, it was fine because there was only some slight sickling to his cells. So I thought that was interesting there because not only does the sickle cell anemia display some incomplete dominance, it also gives some protection against malaria, and that is an example of pleiotropy. Very interesting. All right, so now that he knows that he has that, he can also, be treated for it and knows to be careful when at high altitudes. And so those are two examples of autosomal recessive disorders. That is when the faulty allele is recessive and on an autosome. But the faulty allele isn't always recessive. Sometimes the faulty allele is dominant. And let's go back to blue. Okay, so that's what? 2. I don't want this line here. I'm going to skip over this line. All right, so 2, we have autosomal dominant disorders. This is when the faulty allele is dominant over the recessive. Faulty allele is dominant over the recessive and is on an autosome, which again, an autosome is a non-sex chromosome. Any chromosome that's not a sex chromosome. And an example of this is a type of dwarfism. Not grumpy and sneezy and whatever, but there are several types of dwarfism, and by that I mean people who are short. Peter Dinklage, for example, is a dwarf. I think technically that the definition of someone who has dwarfism is anyone under 5 feet, but that sounds a little ridiculous to me. You're telling me somebody who's 4'11" is considered a dwarf? That's interesting. There are lots of different types of dwarfism, but there is one in particular that displays autosomal dominant inheritance patterns. So one type, and it's the type that is from that old TV show, and they have a spin-off for it now, but it was called Little People, Big World. Little People, Big World. And that's where both parents are dwarves. Yes, that's how you spell it. Dwarves, not dwarfs. They're dwarves. And their kids, oh, they have kids are, they have kids who are dwarfs. I think they have 2 kids who are dwarves and one of normal height. Normal height. This particular type of dwarfism, this is where the dominant allele causes dwarfism. And this time it's the recessive allele that causes normal height. Normal height. So, let's look at the 3 genotypes. Homozygous dominant, heterozygous, and homozygous recessive. Okay, what is the genotype of affected individuals? These are both affected individuals. If you've got the dominant allele, you have the disorder. So these are affected individuals. There is no carrier. Because the thing is, if you carry just one allele, you've got the disorder. Way back here with cystic fibrosis, this person here, up here, a carrier is someone that has the faulty allele but not the disorder. Down here, a person who carries the faulty allele has the disorder. All right. So if we look at these parents that are on Little People, Big World, where both parents are dwarves, but they have kids who are dwarves and they have one kid of normal height, we know that this is the kid of normal height. And so if they have one kid of normal height, what do we know about the parents? We know that they had to get one of those recessive alleles from one parent and one from the other. And we don't know what the other parent's allele is, or do we? Because we know that the parents have dwarfism, so the parents must be heterozygous. All right, give me my blue back. So each parent must be heterozygous. So yeah, when I was watching that show, I was like, oh my gosh, this is a great example for genetics. It displays autosomal dominant disorders. Okay, it's an example of one. All right, so for autosomal dominant and autosomal recessive, you need to know what are the affected genotypes. Right. And otherwise, it's just simple Mendelian inheritance. You know, I mean, if we did this cross here, we did our okay thing like that. We've done that before. All right, and there's a 3 in 1 chance of their kid having the disorder. There's a 1 in 4 chance of their having a kid with normal height. Moving back up here to our notes where we started. Okay, that was autosomal disorders. Now we're going to do X-linked, where the faulty allele is on the X chromosome. Let's scroll back down. So we're on B, X-linked disorders. Faulty allele is on the X chromosome. Inheritance of X-linked disorders is going to be different. And here's why. It's because not everybody has 2 X chromosomes. So all this stuff that we've worked on before, going everybody has 2 factors for the same trait, well, with X-linked disorders, that is not true, okay? Females have 2 shots at getting a healthy allele that's on the X chromosome, but males have only one X chromosome. They got a Y. So inheritance is going to be different. All right? So there is a trait that I'm going to use it as an example here that is called red-green color blindness. Red-green color blindness is caused by a faulty allele on the X chromosome, and in this case the faulty allele is recessive. Caused by a faulty recessive allele on the X chromosome. Okay, so individuals with red-green color blindness, and let's just go with green here. How about a darker green? There we go. Individuals with red-green color blindness cannot see differences between shades of red and shades of green. Mr. Rogers actually was red-green colorblind and couldn't see subtle differences between those shades. And even more so than that, not able to see differences in saturation between different shades of green even. So if you went to school, if you went to elementary schools, let me phrase that differently. If you went to public elementary schools in the U.S., I'll bet you dollars to donuts that you were tested for red-green color blindness. It's when they had you look at these books, and you had to go to the nurse's office or something, or the nurse came to your classroom and they told you, okay, you're going to have a test today, and you're like, they make you look at these books with all these drawings that are like these little bubbles like this, and these bubbles are colored in, and there'll be something like this. I'll let you look this up online if you want, all right? And they'll be like, what number do you see here? And I'm like, the number 2. Was this test supposed to be hard? But individuals with red-green color blindness might not be able to see differences, especially if I shade these in slight shades of green or red right there. Let's erase that. If we were in the laboratory, I could pull out those books because I know where they are. Okay. Alrighty. This faulty allele is on the X chromosome, and so we can't draw things like, and say someone's, this guy's heterozygous for this trait. No. Some people don't have 2 alleles. Only females will have 2 alleles for X-linked traits. Males have only 1 allele for X-linked traits. Let's write that down. Okay. Females have 2 alleles for X-linked traits. Males have only 1 allele for X-linked traits. Because males are XY, females are XX. You may know of someone who has red-green color blindness, or you may have it yourself. Who knows? But just think about it for a moment. Who is more likely to have an X-linked disorder? I'm betting that if you know someone who has red-green color blindness, they are probably male. And that's because X-linked disorders are much more common in males than in females. And that is because females have 2 X chromosomes. So females have 2 chances at getting the healthy allele. Males have only 1 chance at getting the healthy allele. Let's see why. Let's draw our little chart for alleles. Okay. So, well, it's not a chart, not yet. All right, because the allele for red-green color blindness or red-green color vision, let's say, is on the X chromosome, we're not going to, you know, write big R, little r, or something like that. We're going to write the X chromosome, and the healthy allele, which is dominant, will be written as a superscript. So this allele, X with a superscript capital R, causes normal color vision. Whereas the recessive allele, we will write an X to represent the X chromosome, and it's recessive, so the faulty allele is recessive, so we write a lowercase r as a superscript. This causes red-green color blindness. I'll just put CB for color blindness. So we got to make a chart here. There will be more than 3 genotypes that are possible here. There is no R allele. There is no allele for red-green color blindness or red-green color vision on the Y chromosome. So we cannot do stuff like that. No, that doesn't work. None of that. So there we are. So let's make a table now. All right. Genotype in one column, and the phenotype in the next column. So it's entirely possible that an individual could have 2 healthy alleles. Okay, so what is this person's genotype going to be? Yes, they will have normal color vision, but are they male or female? Their sex will be female. So this is a female with normal vision. Another possibility would be a female with one of each. This would be the only true heterozygotes. Okay, this would be a female with normal vision. Right there, but they're a carrier. A carrier is someone who has the faulty allele but not the disorder. So, but a carrier. There we go. All right, there is a third possibility for a female. A female actually could have 2 faulty alleles, so this will be a female with red-green color blindness. All right. I'm not seeing any other options for females, no more combinations there, so let's move on to male. Males are XY. I am in need of tea. There we go. That's much better. Oh, and let's give this guy a dominant allele. So this is a male with normal vision. Yay, people with normal vision can be police officers and can be pilots and can be air traffic controllers. People who are red-green colorblind cannot. You need to be able to see differences between red and green because a lot of the signals, for example, in air traffic control require that you be able to see those differences. So, but not this dude. All right, not this male. But how about this one? Dun dun dun. We have a male with red-green color blindness right there. Okay, so there aren't 3 genotypes here. There are 5. So can you see how it's more likely that males would have a disorder that is X-linked than females? Because think about females right here with this X-linked disorder. I mean, yes, it's entirely possible for a female to have an X-linked disorder, but think about her parents. What do we know about a female? What do we know about the parents of a female with red-green color blindness? Let's take a look at them. Okay, we'll bring them down here. Here's this female way down here with red-green color blindness. We know she had to inherit this from one parent and this other faulty X chromosome from the other one, because she didn't get both of them from the same parent, right? But one of these has to be their birth mother, and the other one, well, let's put the birth father over here. Okay, and our males XX or XY? They're XY. And you cannot write an R on that Y. There is no R there. So for females with red-green color blindness, we know that their father, their birth father, had to have had the disorder. Whereas their mother, their birth mother, they could have had the disorder. You know, we could add the extra X chromosome there. This could be, you know, a dominant allele or a recessive. We don't know, but we would have to know if this female's mother actually had red-green color blindness. But we know that their mother had to have at least been a carrier. Right. Now, several years ago, I was going through this stuff, and one of my students, female students, said, "Oh, my son has red-green color blindness." And we were like, "Oh, isn't that interesting?" Well, let's go with something red for a while. Apologies if you are red-green colorblind and can't see the difference in this color, but I guess you couldn't tell the difference between the green and whatever anyway, or maybe not. But this mother said, "Oh, my son has red-green color blindness." Now, there are a lot of different types of color blindness out there, and she said, "Oh, no, it was definitely diagnosed as red-green color blindness, so we know that that's an X-linked disorder." So we said, "Oh, let's write down your son's phenotype. I'm sorry, your son's genotype." So we're like, okay, he's male, so he's got an X chromosome and a Y chromosome, so which type of R do I put on that X chromosome, the dominant allele or the recessive? And she said, "Oh, well, the recessive allele." So she said, "That's my son right there." I'm like, "Wow, that's really interesting." She's like, "Yeah, he got it from his father because his father's also red-green colorblind." And we were like, "How's that work?" And she said, "Well, his father has red-green color blindness, so our son got his red-green color blindness from his father." And we're like, "Are you sure that he has red-green color blindness?" She said, "Yes, the doctor even said that it's something like an X-linked or sex-linked trait." And we're like, "Okay, let's look at this." We're like, "So from whom did your son inherit his X chromosome?" And she said, "Well, his father." We're like, "Well, if he got his X chromosome from his father, where did he get his Y chromosome? Because it wasn't from his birth mother." We're like, "Whoa." She's like, "No." It was very insistent, and the other students were like, "Oh, no, it doesn't work that way. Your son got his Y chromosome from his father because that's how sex determination works, right?" Here we are. Here's the kid's dad. Here's my former student, the mom. She's got 2 X chromosomes, right? Let me fix this. Because males can only pass on males pass on their Y chromosomes to their son. If they donate a Y chromosome in that sperm, that kid's going to be male, right? It's going to have an X and a Y chromosome. Males inherit their Y chromosomes from their father and their X chromosomes from their mother. And this young woman said he had to have gotten it from his father. His father is red-green color blindness. So everyone in the class, the students all decided that their dad was like that, had a recessive allele on his X chromosome, but he did not give his X chromosome to their son. She's like, "But that would mean that I would." And everyone in the class just laughed because this is her first realization that she said, but I'm not red-green colorblind. So folks, what's the mother's genotype? The mother's genotype is one dominant allele on one of her X chromosomes and a recessive allele on her other. So yes, she did confer. She did pass on the red-green color blindness allele to her son. Males inherit X-linked disorders from their birth mothers. So, inherit X-linked disorders from birth mothers. Males. And that's because males can only pass on X-linked disorders to daughters. Women with X-linked disorders can pass it on to daughters or sons. So let's write that down. Females can pass X-linked traits onto daughters and sons. But males can pass X-linked traits onto only daughters. So that's important. And that is such a different inheritance pattern than autosomal disorders. Now it is possible for a female to have an X-linked disorder. We saw this back here on our little chart right there. This female right there has an X-linked disorder. And what do we know about females with X-linked disorders? From whom did they inherit one X chromosome, and from whom did they inherit the other? One from their mom, and we don't know what this other allele is, and one from their dad, their birth dad and birth mom. Right? So for females with X-linked disorders, we know that their birth father had to have the disorder as well. For females with red-green color blindness, we know that their birth father had to have red-green color blindness as well. And this is actually my sister-in-law, my SIL. My sister-in-law is red-green colorblind, and she cannot see the difference, and it's pretty bad for her too. I mean, like, her significant other has to pick out clothes for her and tell her, like, that top does not go with those pants. We were driving around one time, she's like, oh, these little pink houses are so cute. Or she's like, oh, that's a peachy pink. I'm like, those houses are white. She's like, oh, I can't see the difference. And that's my sister-in-law. Right? Okay. Now, this is my mother-in-law up here. So we know that my father-in-law up here had red-green color blindness. And we know that my mother-in-law had to have at least been a carrier. Now, we don't know, if I change this color here, what that other allele is. Now, when my mother-in-law was alive, she would have insisted that she was not red-green color blindness. However, I have my suspicions because I saw the color that she painted her bathroom. It was neon peach, and I'm not kidding you, it was neon peach. I'm not sure if I have that neon peach up here. It was way more neon than that. Do I even have peach? No, I don't even have peach in my palette here. So, I don't know. She might have been red-green colorblind, but she would insist that she was not. She's not around anymore, so how do we know? Well, we could look at her other children, okay? I'm married to her son. So her son Let's see, he's younger than my sister-in-law, so I'll put him over here. And I can tell you that my husband is not red-green colorblind. So he had to have inherited his X chromosome from his mother. So we know that my mother-in-law must have been heterozygous. So a true heterozygous. She must have been a carrier, but was not red-green colorblind, much to her glee. Or was she? Dun dun dun. And here's our next concept. This is so freaking interesting. And this is something that only happens in females. Now let's say back when my mother-in-law was a single-celled organism, okay? Just a fertilized egg, just a little zygote right here. One cell divides into two, and each of those cells will have an X chromosome with an R, that's a dominant allele, and an X chromosome with the faulty allele, with the recessive allele. And then those two cells will divide, and each of those cells will have one of each, just during regular old mitosis. And then those cells will divide. All right, and I don't know at what cell stage this happens, but at some point early in development, let's go with purple, and this only happens in females. There is a phenomenon called X-inactivation. It's only in females. X-inactivation in females. There is something about having two X chromosomes that makes all these cells go, ooh, we only need one copy of an X chromosome to be okay. Males do it all the time. Males exist just fine with only one X chromosome, and that's the only chromosome for which that works, right? None of the numbered chromosomes, you gotta have two, or the cell freaks out. But the X chromosome, there is something about it, and I don't know if it's the 8-cell stage or the 16-cell stage, but the cell will randomly inactivate one of the X chromosomes, randomly. It'll basically cover it up, shrivel it up. So let's say in this cell here, this one gets shriveled up. But in this one in the middle here, this one gets shriveled up. And just randomly, one of these gets covered up or inactivated in my mother-in-law's cells as when she was at the 8-cell stage. Change the color for a moment here. What if this cell here, this one here, goes on to form the rods and cones in your eyes? The chromosome that's active has the dominant allele on it, so no color blindness. But what if this cell down here were the cell that went on to be the rods and cones, that went on to develop into the rods and cones in my mother-in-law's eyes? She would have been colorblind. Or partially. It is unknown why X-inactivation occurs in females, but it is a phenomenon that happens in all females and not just in humans. It happens in all mammals. On Tuesday, we're going to be doing a cat genetics exercise. And X-inactivation occurs in cats with a gene locus that causes a cat to have orange fur or not. But in females that have one X chromosome with a dominant allele will cause a cat to have orange fur. There is a recessive version, and I'll just draw it like a little dot, causes it to have, basically black fur. It's a little more complex than that. Now, if we substitute these O's for these R's here, Some chromosomes will have this one active, and you'll get patches of orange fur in a cat that is heterozygote for this, and black fur. So they're actually called tortie or tortoise shell cats, and also calico cats. Calico cats are actually tortoiseshell cats that have white tummies too, but we will see this on Tuesday. And it is due to X-inactivation. And that's all I have for you today. I hope you have taken notes on this. You can certainly come back and look at it, but this is going to make things go a whole lot easier for us on Tuesday, so I will see you there.